In the frigid darkness of a remote cave deep within the Altai Mountains of Siberia, a team of scientists from the Max Planck Institute for Evolutionary Anthropology, led by Professor Svante Pääbo, carefully sifted through layers of ancient soil and fractured bones buried for tens of thousands of years. Among the rubble, they discovered a tiny fragment of bone, no larger than half a human fingertip. At first glance, it seemed insignificant, just another relic of a long-lost past. But hidden within this small piece of bone was a secret that would challenge everything we thought we knew about our origins. When its ancient DNA was finally decoded, it revealed a shocking truth. Mysterious human species once lived alongside and even interbred with our ancestors. Could traces of these ancient beings still exist within you today? Like, comment, and subscribe as we embark on this journey into our shared past. For centuries, scientists have pieced together the story of human evolution using fossils and archaeological discoveries. According to the traditional theory, Homo sapiens, modern humans, emerged around 300,000 years ago in Africa. From there, they began long migrations across continents, gradually spreading into Europe and Asia, eventually replacing earlier human species that had existed before them. This model was built on landmark discoveries. In 1856, the first Neanderthal fossil was unearthed in the Neander Valley, Germany, offering the first glimpse of a close relative of modern humans. Later, in the early 20th century, the pioneering Leakey family, including Louis and Mary Leakey, uncovered remarkable fossils in Olduvai Gorge, Tanzania, identifying early species like Homo habilis and Homo erectus. These finds seem to support a straightforward view of evolution, one species emerging after another in a linear progression toward modern humans. Other iconic sites, like La Chapelle-aux-Saints in France, revealed nearly complete Neanderthal skeletons, while discoveries in Dmanisi, Georgia, provided crucial evidence of Homo erectus migrating out of Africa. These fossils became the foundation for the belief that Homo sapiens were superior, smarter, more adaptable, and that this superiority allowed them to outcompete and ultimately replace other species entirely. This traditional view was represented as a simple evolutionary tree, where each branch that emerged replaced the one before it. It painted a clear, orderly picture that appeared in the textbooks, museums, and popular culture. Yet, even as this narrative took root, there remain puzzling gaps, missing connections and unexplained patterns in the fossil record. These silent spaces hinted at a far more complex and mysterious history, suggesting that the true story of our origins was far richer and far more tangled than the tidy model we had long believed. In 2008, deep inside Denisova Cave in the Altai Mountains of Siberia, Russian archaeologists unearthed a small, unremarkable bone fragment. At first, it appeared to be nothing extraordinary, merely another piece of ancient debris among countless others. The fragment was sent to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where it came under the careful study of Professor Svante Pääbo and his team, pioneers in the field of ancient DNA research. Initially, the scientists assumed it belonged to a Neanderthal, a close relative of modern humans already known to have lived in the region. But when the team extracted and sequenced its DNA, the results were completely unexpected. Published in the journal Nature in 2010, their analysis revealed an entirely distinct lineage of humans, previously unknown to science. This mysterious group was given the name Denisovans, after the cave where the fragment had been found. The discovery was nothing short of groundbreaking. For decades, the prevailing model had suggested that the story of human evolution was relatively simple. Homo sapiens expanded from Africa, replacing other species like Neanderthals and Homo erectus. But the Denisovan DNA told a different story, one far more complex and surprising. As Pääbo's team compared the Denisovan genome to modern human populations, they made an even more astonishing revelation. Living people today still carry Denisovan DNA. Populations in Melanesia, Papua New Guinea, and parts of Southeast Asia were found to have up to 5% of their genome inherited from this mysterious species. Even more intriguingly, Tibetans were discovered to possess a unique gene variant called EPAS1, which enables them to thrive at extremely high altitudes. This gene was traced directly back to Denisovans, revealing that their genetic legacy continues to shape human survival today. These findings suggested that rather than replacing other species entirely, Homo sapiens interbred with them, creating a complex web of genetic exchange. The clean linear image of evolution, one species triumphing over another, was shattered. Instead, humanity's past appeared more like a tangled network, with ancient species interacting, mixing, and leaving behind genetic echoes that persist to this day. The Denisovan discovery didn't just add another branch to the human family tree. It completely rewrote our understanding of what it means to be human, opening the door to new questions about who we are, where we came from, and what other secrets might still be buried beneath the earth, waiting to be revealed. The discovery of the Denisovans did more than reveal a mysterious new branch of the human family tree. It also pushed the boundaries of what science could achieve, demonstrating the power of ancient DNA analysis to uncover secrets hidden for tens of thousands of years. To truly understand the significance of this breakthrough, we must journey inside the laboratories where these fragments of the past are painstakingly transformed into genetic revelations. When a bone or tooth fragment is discovered, the first and most critical step is preservation and contamination prevention. Ancient DNA is extremely fragile, often broken into tiny pieces by time, heat, moisture, and bacteria. Even a single particle of modern human DNA from a scientist's skin cell could completely overwhelm the ancient genetic material. That is why the labs at the Max Planck Institute for Evolutionary Anthropology, led by Professor Svante Pääbo, are designed like biological clean rooms, similar to those used for building spacecraft. Scientists wear full-body suits, gloves, and face masks. The rooms are pressurized, and every surface is sterilized constantly. Once a sample is prepared, researchers carefully drill into the bone, extracting a fine powder. This powder is treated with specialized chemicals to release any remaining fragments of DNA. The next step involves purifying the DNA, isolating it from dirt, proteins, and contaminants. Because ancient DNA is so degraded, scientists use advanced techniques to repair and amplify the fragments, making them readable for sequencing machines. The breakthrough came with the rise of next-generation sequencing, NGS, a technology that allows millions of fragments to be read simultaneously. This innovation revolutionized the field of paleogenetics, enabling scientists to piece together entire genomes from even the most damaged samples. The process is a bit like assembling a massive puzzle with millions of tiny pieces, most of which are missing or damaged, and trying to reconstruct the complete picture. At Max Planck, Pääbo's team compared the Denisovan genome to both Neanderthals and modern humans. The results revealed not only how Denisovans were genetically distinct, but also when and where they might have interacted with other species. Their studies showed that Denisovans and Neanderthals shared a common ancestor, splitting from each other around 400,000 to 500,000 years ago. Later, as Homo sapiens migrated out of Africa, they encountered both of these species, and instead of simply replacing them, they interbred. One of the most striking examples of this genetic legacy involves a single gene, EPAS1. In 2014, a team led by Rasmus Nielsen at the University of California, Berkeley, and Jacob Crawford discovered that Tibetans possess a unique variant of EPAS1 that helps them survive in oxygen-poor, high-altitude environments like the Himalayas. By comparing it to ancient genomes, scientists determined that this gene was inherited directly from Denisovans. This finding provided a vivid example of how ancient DNA continues to shape modern human biology, even today. The implications extend far beyond Tibet. Denisovan DNA has been identified in populations across Southeast Asia, including Papua New Guinea and parts of Indonesia, suggesting that these ancient humans were once widespread throughout the region. Similarly, studies have revealed that modern Europeans and Asians carry Neanderthal DNA, which influences traits ranging from immune system responses to hair and skin characteristics. These discoveries have transformed our understanding of human evolution. Rather than a simple narrative of one species triumphing over another, the genetic evidence paints a picture of a complex network of interactions, with different human groups meeting, mingling, and leaving behind fragments of themselves in our DNA. This tangled web challenges the idea of clear boundaries between species, revealing instead that the story of humanity is one of shared heritage and intertwined destinies. Visually, it is almost cinematic. Imagine a darkened lab illuminated by the glow of computer screens, where a researcher watches as lines of genetic code scroll across the monitor. Somewhere in that data lies the trace of a being who lived and died tens of thousands of years ago, a being whose existence was unknown until this very moment. The realization is profound. The past is not truly gone, for its echoes still flow in our veins. Through painstaking work, these scientists have not only uncovered a new species, but also demonstrated how cutting-edge technology can breathe life into ancient mysteries. With every fragment analyzed and every genome sequenced, they bring us closer to understanding the true origins of humankind and the other human species that still live on inside each of us. For years, the discovery of Denisovans had fascinated scientists, but in 2018, an extraordinary find would take the mystery to an entirely new level. While examining thousands of bone fragments from Denisova Cave, a researcher from the Max Planck Institute for Evolutionary Anthropology, Vivian Sloan, came across a tiny, weathered piece of bone about the size of a thumbnail. At first glance, it seemed unremarkable, much like the fragment that had first revealed the existence of Denisovans a decade earlier. When the team extracted DNA from this bone and began sequencing it, they were stunned by what they saw. The results showed something unprecedented in the history of paleoanthropology. The individual to whom the bone once belonged had a Neanderthal mother and a Denisovan father. This was not just evidence of two species coexisting. It was direct proof of interbreeding, a tangible connection between two ancient human lineages that had long been thought of as separate. Published in the journal Nature later that year, this revelation electrified the scientific community. For decades, researchers had speculated that interbreeding must have occurred because fragments of Neanderthal and Denisovan DNA are found in modern humans. But this single bone, belonging to a young female nicknamed Denny by the researchers, was the smoking gun they had been searching for. She was the first confirmed hybrid ever discovered, a literal child of two worlds that were thought to be rivals. The genetic data provided even more fascinating details. Denny's mother, a Neanderthal, was closely related to a group of Neanderthals living thousands of miles away in Western Europe, suggesting long-distance migrations and interconnections between populations. Her father, a Denisovan, also carried traces of Neanderthal ancestry, revealing that these encounters between groups were not isolated events, but part of a complex web of relationships spanning millennia. This single fragment of bone demolished the old simplistic vision of human evolution as a straight line. It revealed a world where different species of humans not only met but lived together, shared knowledge, formed bonds, and even raised children. In that moment, the boundaries between us and them blurred forever, and humanity's past became more intricate and deeply interconnected than anyone had ever imagined. As the team stared at the data, they realized they weren't just looking at the genetic code of an extinct species. They were witnessing a moment of love, survival, and union preserved in the fragile language of DNA. The discovery of Denny, the hybrid child of a Neanderthal mother and a Denisovan father, profoundly reshaped how we think about human evolution. It proved beyond doubt that our past was not defined by isolation or competition alone, but by connection, exchange, and shared survival. This revelation forced scientists and historians alike to reconsider what it truly means to be human. For decades, the dominant image of evolution was a straight ladder, with each species replacing the one before it. But the genetic evidence revealed by ancient DNA paints a far richer and more tangled picture. Instead of a ladder, human evolution resembles a web of intertwining branches, where different species met, mingled, and left lasting marks on each other. We are not the pure descendants of a single lineage. We are a mosaic of many ancient peoples, their stories still written within our very cells. The Denisovan contribution to modern humans illustrates this beautifully. In Tibet, the EPAS1 gene, inherited from Denisovans, allows people to survive at extreme altitudes where oxygen is scarce. Without this ancient gift, it's possible that entire populations could never have thrived in such harsh environments. Similarly, Neanderthal DNA influences the way our immune systems fight disease, our skin adapts to sunlight, and even how our bodies process certain nutrients. These fragments of ancient code are not relics of the past. They shape our daily lives in the present. Beyond science, this discovery carries a profound philosophical and emotional weight. It tells us that we are deeply connected to those who came before us, even to species long extinct. The boundaries between us and them dissolve when we realize that their survival struggles, their triumphs, and even their love stories are woven into who we are today. In a sense, every person alive is a living time capsule. Our blood carries echoes of vanished worlds, a record of migrations, encounters, and shared destinies stretching back hundreds of thousands of years. Ancient DNA is more than a scientific tool. It is a bridge across time, allowing us to see not just fossils and artifacts, but the living legacy of our extraordinary interconnected past. As the last rays of sunlight fade over the snow-covered Altai Mountains, one can almost imagine a distant past where fires once burned inside Denisova Cave. Around those ancient flames, Neanderthals, Denisovans, and perhaps even early Homo sapiens huddled together, sharing warmth and stories we will never hear. Today, their voices are silent, their bodies long gone, but their presence lives on within us, encoded in the intricate patterns of our DNA. The discoveries made by scientists like Svante Pääbo, Vivian Sloan, and their teams remind us that human history is not a story of separation, but of connection. We are not simply descendants of one species replacing another. We are the living outcome of countless encounters, migrations, and unions that shaped who we are. When you look in the mirror, you are seeing more than just yourself. You are seeing a tapestry woven from countless threads, a genetic legacy stretching back to a time when the world was young and mysterious. If this journey through deep time has inspired you, consider liking this video, sharing it with friends, and subscribing to our channel. Together, let us continue exploring the secrets hidden in our shared human story.